Good afternoon. I welcome you all to the online parallel session 2 on environmental sciences. This session contains 7 scientific presentations. Environmental science session chaired by Professor BCL Athapattu, Banduni Athapattu. She is director of the Center for Environmental Studies and Sustainable Development. Also, she is a professor in environmental engineering in Department of Civil Engineering, Open University of Sri Lanka. So, over to you, madam.
Thank you very much, Dr. Gihan Dahanayaka, who is the co-chairperson of this session. Welcome all of you for the session of environmental sciences. I hope that all the authors know that all the presenters know that it's 10 minutes presentation followed by 5 minutes question and answers or the discussion session. Right, all of you very welcome to the IAWS International Open University Research Sessions in 2022. We will start our first presentation. The first presentation is ID number ID number 13. The title of the presentation is Impact of Shrimp Farming on Degradation of Diversity, Soil and Water Quality of Mangrove Ecosystem in Anavilundava Ramsar Wetland Sanctuary. The presenters, the authors are HMSM Bandara, WLBP Upananda, and CS De Silva. So we welcome all of you, and forum is over to you for the presentation. The research is presented by HMSM Bandara. Over to you.
Good afternoon, all of you. I am HMSM Bandara. My research is Impact of Shrimp Farming on Degradation of Diversity, Soil and Water Quality of Mangrove Ecosystem in Anavilundava International Ramsar Wetland Sanctuary. Introduction. Mangroves are among the most productive ecosystems in the world and coastal equivalent of tropical forest and hence of important ecological and environmental significance in Sri Lanka. It is estimated that mangroves in Sri Lanka cover only 160 square kilometers. The largest tract of mangrove habitats in Sri Lanka are found in Puttalam Lagoon, Kalaoe Basin and Trincomalee. Mangroves provide many ecosystem products and services. The mangrove forests in the Puttalam district have suffered the highest level of degradation arising from human activities, particularly shrimp farming. The direct destruction of mangroves, shrimp farming has also caused the degradation of water quality and the loss of soil characteristics. This experiment we can identify the impact of shrimp farming on mangrove ecosystem and determine how to minimize loss in mangrove ecosystem in Anavilundava International Wetland Sanctuary. The objective of this study to determine impact of shrimp farming on diversity, soil and water quality of mangrove ecosystem of Anavilundava International Ramsar Wetland Sanctuary. Methodology. Study sites. 3 mangrove sites were selected in this area. Selected mangrove sites were as follows: Pinkattiya area, Aanvilundawa area and Singalloya area. Experiment 1. Impacts were monitored in 3 mangrove sites located adjacent to shrimp farms that were currently in operation, while 3 sites away from shrimp farm and soil and water samples were taken as below. Experiment 2. Survey was conducted to identify the true mangrove species in Aanvilundawa International Ramsar Wetland Sanctuary. In this survey that was conducted according to the guidebook Mangroves of Sri Lanka, related to the morphological characters of the species found in the respective sections by using a rope 20 meter by 20 meter along the Dutch Canal. Experiment design. The experiment was set up as 2-factor factorial randomized complete block design with 4 replicates. Data analysis. Each parameter were analyzed using ANOVA procedures of SAS. Means will be compared using Turkey test. Results and discussion. Water temperature. Water temperature measured from close and distant locations of shrimp farm in Singalloya and Aanvilundawa significantly varied, but in the water temperature of Pinkattiya there is no significant differences between away and near sites of shrimp farm. Highest water temperature recorded in Aanvilundawa site. Soil temperature. Highest soil temperature was recorded near the shrimp farm in Aanvilundawa site and lowest soil temperature was away from the shrimp farm in Aanvilundawa. Water pH. Highest water pH level was recorded from near the shrimp farm of Aanvilundawa site, as well as lowest pH level showed in a location distant from the shrimp farm of Singalloya site. Soil pH. The highest soil pH level was shown in a place near the shrimp farm in Aanvilundawa site, as well as a distant place from the shrimp farm at pH level was recorded and it was significantly different. Water salinity. Water salinity levels of all sites varied significantly. Soil salinity levels of all sites were significantly different as in the case of water salinity level. Water total nitrogen percentage. There was a significant difference between the total nitrogen percentage of water in Singalloya and Aanvilundawa sites. The total nitrogen percentage of soil was significantly changed between 2 places of sample collect in all sites. Water total phosphorus percentage. There was a significant difference of total phosphorus percentage of water in Singalloya and Aanvilundawa, but not in Pinkattiya site. This research proved that statement because most of the water and soil parameters were significantly changed near shrimp areas. Also, both soil and water total nitrogen percentage as well as total phosphorus percentage were significantly increased near the shrimp farms. Furthermore, research needed to define what are the reasons for increased total nitrogen levels and total phosphorus levels in the soil and water near the shrimp farm because farmers are using various types of chemicals and feeds. A survey was conducted to identify the true mangrove species in Anavilundava Ramsar Wetland Sanctuary. The National Red List 2012 completed by Biodiversity Secretariat 22 species of true mangrove in Sri Lanka. Of the 22 mangrove species in Sri Lanka, but only 8 true mangrove species and 4 species are mangrove associate found in the Anavilundava Ramsar Wetland Sanctuary. Conclusions. The present study shown that rapid growth of shrimp farms in this area has serious negative impacts on mangrove ecosystem in Anavilundava International Ramsar Wetland Sanctuary. This was evident through its impact on both soil and water in this area. The composition of soil and water were seriously altered in the vicinity of shrimp farm in comparison to that of the away and near sites. Despite the constant flushing water quality of the Olanda Ala has deteriorated as apparent by the greater levels of nutrients implementation of earthification and the potential toxicity of mangroves. References. And thank you all of you.
Thank you very much, Ms. Bandara, for your nice presentation. Now the forum is over to the audience for questioning and answering sessions. You can forward your questions. Please unmute and raise your question. If there are no questions, may I ask question from Ms. Bandara? Would you please let us know that the phosphorus and nitrogen level in the shrimp within the shrimp farm? Have you got this data? Have you collected that data? Because you have got the soil and water near the shrimp farms. Can we know that the data or any soil and any nitrogen and phosphorus level within the shrimp farms? Ms. Bandaro, any of the authors can answer for the question.
Yeah, I'm Professor Shanti here. So they have not done within the stream farm because they cannot approach that area. I think they have done near that.
Okay, thank you. So you don't have any idea of because of that the levels within the farms?
Yeah, within the farm.
Yeah,
they haven't taken the analysis.
Right. Okay. So the impacts of mangroves, how you are presenting? I mean that we know that nitrogen phosphorus are really good for the mangroves. It's a plant nutrients. So therefore that impacts are coming through the toxics or any other things. How do you define that, or how do you give the explanation regarding that? Ms. Bandara? May I get the answer from anybody? Or
Yeah, yeah. The thing is, I think they have seen the impact of shrimp farming on mangrove species, and they also found that mangrove species have been decreased compared to the earlier one. So there's an effect on that.
Okay. Any other questions? It's really important research, because that it's really good that we know that how near and far the water quality impacts with these shrimp farms. We know that it's really bother with the eutrophication and all. And so, Ms. Bandara, do you have any future aspects or in future research based on this? Are you going to have any research based on this, or expand your research in future?
Yes.
Yes. What kind of research? Or you'd like to know your future plans regarding this research? How do you carry on this in future?
I'm still working wildlife conservation of Sri Lanka, this area, and
Okay.
start new research this area, madam.
Okay. Right. Thank you very much. Shall we move to the next session? Okay.
Thank you.
Thank you. Thank you for all the authors and contributing to the IAVA's international session. Thank you. We will move to the next session. That is ID number 132. The title of the presentation is Impact of X-ray Radiation. Impact of X-ray. What is it? I'm sorry. Yes. The next one is ID number 132, application of INVEST model to evaluate blue carbon dynamic. Extremely sorry that.
Madam, my number is 96. It's the effects of X radiation.
Yes,
96.
I think I was reading the first at the correct one. It's mixed up. Yes. Extremely sorry. Okay. Again, I'm recalling that the ID number 96. The title of the presentation is Effect of X-ray Irradiation on Seed Germination and Seedling Growth of Indian Gooseberry. The authors are WPAM Vijay Singh and SNCM Dias. The forum is over to the presenter, WPAM Vijay Singh. Thank you.
Good afternoon, everyone. I'm WPAM Vijay Singh, final year MSc candidate from the Department of Environmental Science, CESSD, Open University of Sri Lanka. My research is to find out the effect of X-ray irradiation on seed germination and seedling growth of Indian gooseberry, Phyllanthus amblica. My research supervisor is Dr. Mrs. SNCM Dias. She is a senior lecturer and a research coordinator, CESSD, OUSL. As my content, first I would like to give brief introduction of the study, then the methodology, results and discussion as a third section, and the conclusion will be explained as a fourth part. Finally, my presentation will be ended up with references. The selected study crop, Phyllanthus amblica, is an essential medicinal plant in traditional medicine. The whole parts of the plant contains huge nutritional value, such as micronutrients, but it's still categorized as an unutilized fruit species due to insufficient availability of quality planting materials. Phyllanthus amblica seeds exhibits long-term dormancy by hindering seeds propagation potential. The existing propagation methods are highly technical and expensive, and some methods causes to damage existing plant population. As the aim of the study was needed to find out an artificial methods to break up seeds dormancy in Phyllanthus umbilica. Low-dose X radiation was selected as an artificial method in this experiment. The study was investigated whether the radiation technology can be applied as a low-cost, feasible, and efficient seed treatment methods to break up seed dormancy to increase seeds propagation potential without damaging existing plant population. The study was targeted to obtain quality planting materials to promote large-scale cultivation methods, fulfill the demand within the country. Let me explain the methodology. Selecting viable seed sample is the initial step. Firstly, we sorted apparently quality seeds by observing through hand lens, and then transferred to floating test. The seeds sank to the bottom were selected as viable seeds for further steps. The selected seeds were soaked in water beaker for 20 hours and seed surfaces were sterilized using 1% sodium hypochlorite solution to prevent fungal infection during the testing period. Then the prepared seed sample was placed in labeled petri dishes according to the dose values and replications. After that, the arranged seeds plates were exposed to the radiation according to the each dose value by using computed tomography 126 slice equipment. The irradiated sample was stored in mini greenhouse for germination. Number of germinated seeds and seedling growth parameters were measured using calibrated measuring devices. MS Excel 2010 was used for descriptive statistical analysis and graphical data representation. This study was conducted as a quantitative study. The exposure treatment has been performed at Asiri Hospital, Colombo 5, and the rest of the procedures and mini greenhouse was located in Vallampitiya, Colombo district in Sri Lanka. Prevailing weather condition was affected as an external factor during the study. The experiment was designed as randomized complete block design and used single variable factor. The factor is X-ray dose. One-way ANOVA was carried out to find out the significant difference of the findings. Here we selected 0 is the control to 7.5 gray X-ray dose range with 0.5 gray intervals. The test was selected 16 doses and replicated 6 times per each dose level. Data were collected every week after treatment within 3 months period. Finally, 96 datasets were analyzed for variance and least significant variance at 0.05 standard deviation was used in analysis process. Final germination percentage, mean growth time, vigor index were also calculated according to the equations below to identify the germination process. Let me explain the summary of the results. A significant higher seed germination percentage was obtained in 2.0 gray exposed plate, when the significant higher seedling growth was obtained in 1.0 gray dose level. In between 1.0 and 3.0 gray dose level, treated sample noted a significant higher germination percentage. In between 0.5 and 4.0 gray treated sample showed a significant higher seedling height of the study. The dose level above 4.5 gray were exhibited the detrimental and inhibitory effects in germination process. The significant higher seeds vigor index and fastest mean germination time was obtained in 1.5 gray dose exposed sample. These two line charts are expressed in detailed picture in seed germination percentage and seedling growth in Phyllanthus umbilica as I summarized in previous slides. As a limitation of the study, we used long-living study crop for short-term experiment. The time period was limited to study the total effect of radiation in different growth stages of this species, such as flowering, fruiting, like stages, and effects of wheel growth and susceptibility of diseases. The second limitation factor is environmental condition. The study crop's native habitat is based on dry and intermediate zone, but we tested it in wet-based environment. It can be an effect as an external effect to this study. Okay, let's move on to the final part of the presentation. We can suggest the seed treatment method as feasible and quality seed treatment methods to improve seeds propagation in Phyllanthus amblica. The seed treatment methods open up a pathway to obtain required consumption demand through large-scale cultivation by promoting seeds propagation and different underutilized crops. When considered the effects of radiation, there can be identified one in stimulatory effect, which induces physiological process, and the other one is detrimental or inhibitory effect, which causes genetical and the physiological damages in a species. We have observed these both effects as our findings. In recommendation, we can strongly suggest that the seeds treatment method as a suitable seed treatment technique for Phyllanthus amblica. This method can be developed to obtain feasible and cost-effective large-scale cultivation to fulfill the required nutritional demand within the country. We can suggest the seed treatment methods to apply for other underutilized crop varieties to obtain quality planted materials to develop large-scale cultivation. But before apply this process, we need to find out each suitable dose range which stimulate germination in species. The effective dose will be different due to species diversity. These are my references, and thank you very much for listening to my presentation.
Thank you very much, Ms. W.P.A.M. Vijayasinghe, for the nice presentation. And it's very novel research as far as I know, I considered. And so would you please let us, okay, first of all, let's give the opportunity to the audience to raise questions. We'll be given 5 minutes to questions and answering session. Any questions from the audience? Please unmute your microphone and ask questions. If there are no questions, may I raise a question? Ms. Vijayasinghe, may I know that whether your method is cost effective? Because we know that X-ray radiation or applying of X-ray, anyway, the usage is really difficult because we know that it could be under the control condition. So would you please let us know that whether it's a cost effective?
Yes, madam. I have done the feasibility study by comparing with the other existing procedures, such as the chemical scarification methods, the tissue culture method, and the gibberellic acid treatment method, and other scarification methods of the seed squirts. I have done the feasibility study by taking the analysis plan of the designing and the technology peoples to cost of for the service and all combining. According to that, I got the highest feasibility and highest benefit cost analysis in X-ray treatment. Here, the thing is, I suggest this to as a further treatment. I wish to improve this treatment as a seed treatment technique for other crop varieties also. Here I plan to use the used X-ray devices in medical used X-ray devices. So when we compare with the other radiation in gamma and alpha, beta, like in most experiments has done using gamma radiation. When we compare the gamma radiation, there must be a high protective environment because high penetrability and the energy of the gamma ray. So in X-ray, here we're not using the radiation source like in gamma ray. In gamma, they are using the cobalt 60 like sources. It cannot prevent the emission of the radiation. But in X-ray, we are using the electronic device so to generate X-ray. So we can prevent it easily. if we use the used extra machine and the environment, it is really feasible and very low cost will be taken for that. According to the prevailing economical condition of the country, we can't purchase for hire to take the new machines. So I suggest to use used medical equipments, but it's really can use, but the used medical equipments we can use for seed treatment technique.
Okay, thank you. And so is this the first time that we are using this X-ray radiation or irradiation for this kind of seed germination? Are they used for some other kind of seeds?
Yes, madam. According to my research reveals, I have seen they are in Arab. They have find seed X-radiation effects in seed germination of date palm seeds, and they obtain the seedling growth and the leaf growth stimulation according to the using of low dose X-radiation. So that was based on my experiment also. But they have never. This is the first experiment have seen in Phyllanthus zamblica or underutilized root crop. No other experience has done for underutilized crops or to break up seed dormancy.
I think this fruit is something means like kind of a special for Sri Lanka or some other tropical countries. It's not happening in the other countries, isn't it? This particular,
yes, madam. This is based on tropical climate country. This is based on tropical climate country, not only Sri Lanka. So it is. It can, but the productions of the Indian gooseberry, like in hair dyes, and a lot of productions they are doing using Phyllanthus zamblica. So that's why I selected this. But it's not only in Sri Lanka, but in basically in tropical climate country, Uzbekistan, Pakistan, China also they have. That's
okay. Thank you. One more question that because we know that this Indian gooseberry seed is really hard. So that's why you have selected this X-ray X-ray irradiation method, or any other specialty or any other advantages or disadvantages. Rather than the disadvantages, I'm asking about the advantages of using this particular method of this Indian gooseberry seeds.
Yes, madam. I have noted the mechanical dormancy in the seed coat dormancy in Phyllanthus zamblica due to the hard seed coat, and there's a seed coat dormancy. And another thing is, according to the existing methods of breakup dormancy in Phyllanthus zamblica, is a little bit higher cost than this method, and that is one reason. And another reason is to this is the basic thing is actually it contains dormancy, and I searched the underutilized crop with dormancy. So then I selected this crop because underutilized fruits, they have a lot of nutrient values and medicinal value. So that's why I selected this crop. And the method I used because of my background and my knowledge of irradiation. So I wish to design a new technology to treat seeds other than the existing methods.
Okay. Thank you very much, Ms. Vijay Singh, as well as Ms. Dias. Congratulations that, for your research and
Thank
also
you,
future work. Thank you.
Thank you very much, Mr. Thank you.
Thank you, madam.
Let's move to the next presentation. The third presentation of our session, Environmental Science. The ID number is 132, ID 132. The topic is application of INVEST model to evaluate blue carbon dynamics in the Chilaw Lagoon, Sri Lanka. The authors are O.W. Kotagama, K.A.R.S. Pereira, D.D.G.L. Dahanayaka, S. Somaratna, and S. Patirage. Now the presenter is Mr. O.W. Kotagama. Mr. Kotagama, the forum is over to you.
Good morning, everybody. My name is Odath Kotagama, and I will be presenting on the application of the INVEST model to evaluate blue carbon dynamics in the Chilaw Lagoon of Sri Lanka. Before we get into the study itself, first just a brief description on what blue carbon ecosystems are and the impacts that rising sea levels could potentially have on them. Blue carbon ecosystems is a general term which we use to refer to mangrove salt marshes and seagrass beds. The reason we call them blue carbon ecosystems is one, because they are associated with marine aquatic environments, hence blue, And carbon, because they are known to store very large quantities of carbon, the capacity for these ecosystems to store carbon is found to be even greater than what we observe in terrestrial rainforests. So for a long time, we thought that terrestrial rainforests were the ecosystems that had the potential to capture as much carbon from the environment and mitigate climate change. But now we have found that these blue carbon ecosystems actually have a much larger capacity to do so. But of course, when we look at the distribution, blue carbon ecosystems are much more restricted in their distribution in comparison to terrestrial rainforests. Rising sea levels, which is an impact of climate change, can have a potential impact on blue carbon ecosystems and their capacity to capture this carbon and function in this manner because of their proximity to the ocean and also because of the impacts that inundation could potentially have on them. So with that in mind, in this study what we try to do is to understand what the impacts of rising sea levels could be on the blue carbon dynamics of a lagoon in Sri Lanka, and for this purpose we selected the Chilau Lagoon. And as the objective tells you, we made use of a model that is known as the INVEST model. So the INVEST model is an open source model. INVEST stands for Integrated Valuation of Ecosystem Services and Trade-offs. And in essence, the model is to be used, the model is actually developed to be used to evaluate the trade-offs that happen with different types of management policies that are placed on ecosystems. So if we have an ecosystem and we want to manage it in a particular way, and we have all these different options of managing it, when we do so, what are the trade-offs between the ecosystem services that are present in that ecosystem that can happen under each of those management scenarios. But for our study, rather than making use of management scenarios, we made use of these inundation scenarios, and using this a particular tool in the INVEST model, which is known as the coastal blue carbon tool, we tried to determine the blue carbon dynamics. So the methodology, it's quite straightforward. First of all, we had to obtain information from the Survey General Department, and we had to develop land use maps for the Chilau Lagoon, and we did that making use of ArcMap. Once we had developed the land use maps, which had been updated to the present scenarios, we then determined the extents of inundation with each of the sea level rise scenarios. The different sea level rise scenarios which we took into consideration are shown here. So we made use of the predictions that were given by IPCC in 2021, so with AR6, and that we considered two main scenarios, the very low GHG emission scenario and the very high GHG emission scenarios. And for each of those, we see that there are different inundation extents that are given for the years to 100 and to 150. So the low GHG emission scenario is the very best pathway that could come about given the global trajectories of climate change, and the very high GHG emission scenario is the worst possible outcome which could take place. So once we had identified the different extents of inundation, now we needed to identify how the potential impacts of sea level rise could affect the different types of land use classes around the Chilau Lagoon. So through the initial land use map which we had developed, we were able to identify the different types of land use classes. And now we needed to make use of those DEMs which we had created earlier to identify which of those land use classes are being subjected to inundation and could potentially be affected by sea level rise. So for that, we developed first DEMs using IDW interpolation methods, and these are digital elevation models. The digital elevation model would show us the impacts of rising sea levels and which areas of the lagoon would be subjected to inundation if the sea levels rose to a particular level. Those levels were what we indicated in the scenarios earlier. So the DEMs, once we obtained, we reclassified them based on those scenarios, and we identified the extents of inundation. By overlaying this on the developed land use map which we formed earlier, we could identify which of the different types of land use classes are being affected by rising sea levels. This we then converted into raster maps and introduced into the INVEST model. The INVEST model relies on two forms of input, one being these raster maps and the second being data regarding different types of carbon pools. And three main carbon pools are taken into consideration: litter, soil carbon, and biomass carbon. Those information we obtained from secondary data sources. So the combination of these, we ran the model and we were able to determine what sort of changes could potentially take place as a result of rising sea levels. When we look at the results, Here we have shown the different maps which we have generated making use of the inundation extents. So this is the combination of the land use maps and the digital elevation models which indicate the extents of inundation. Inundation areas are shown in red. This is for the first scenario, which is the best possible outcome with climate change. In these two maps, we can observe the impacts of rising sea levels under the fourth scenario, which is the worst possible outcome for climate change, that is under very high GHG emissions. Particularly, you can see in the years 2150 how the extents of inundation are far more extensive than what we observed in the previous maps. These are the outputs which are generated from the INVEST model. Making use of these, we can then determine the different criteria for the carbon dynamics. Here we can observe the outcomes of the INVEST model, and in relation to the blue carbon dynamics, we have considered 5 criteria: carbon accumulation, carbon stocks, carbon emissions, carbon sequestration, and carbon valuation. We can see then in the year for the carbon accumulation for the year 2021 to 2100, the outcomes are the same. This is because the impacts of sea level rise become more distinctive from the years of 2100 onwards. But between the period of 2100 to 2150, in scenario 4, there is a distinctive decline in carbon accumulation. The same is observed in the carbon stocks, where in 2150, the stock of carbon that is observed at that point is less than what we in scenario 4 is less than what we observe in scenario 1. In the case of carbon emissions, however, there is a distinctive increase in the worst possible outcome in scenario 4. This is expected because what we believe will happen is with the rising sea level and the amount of disturbance which takes place, carbon which is stored within the soils, carbon within the plants, they are going to be released. Therefore, emissions are predicted to increase under the worst possible outcome of climate change and rising sea levels. Carbon sequestration and carbon valuation follow a similar trend, where in the worst scenario, or in scenario 4, there is a decline in the criteria which we are looking at. So carbon sequestration, we see there is a decline in it, as well as in the case of valuation, you see that the prices are much lower in the case of the worst possible outcome for rising sea levels. So in conclusion, and just to draw upon some closing remarks which have been pervasive throughout this study, the INVEST model is extremely beneficial to us because it gives us this sort of predictive information. So the model tells us that based on the scenarios of rising sea levels, carbon stocks accumulation, as well as carbon sequestration and carbon valuation, all would potentially decline if we were to go towards trajectories representing the worst possible scenarios, whereas emissions would increase. And each of these have their own particular implications. These types of predictive information, it's extremely beneficial to us, especially because we are generating a foundation to which we can work on. It's very difficult for us to understand the impacts of climate change, especially because they are so complex, and also because we don't necessarily know how the situation on the ground, the scenario on the ground will be in the next 100 years, next 200 years. Predictive information in that sense is extremely beneficial because it can give us a gist of what could potentially happen. Once we know what could potentially happen, it's up to us to make the decisions which are based on those particular outcomes. But it gives us something to work on rather than working blindly towards these conservation and management practices. Apart from that, the nature of the open source model is also beneficial because most of the technologies and capacities that we have are not readily available to us. They are either very expensive, or we don't have the technical capacity to run these types of models. So the INVEST model, being an open source, it's an extremely good candidate if you were to build up on studies in other different lagoons of the country, or even make use of the other tools available under the INVEST model to look at these sort of changes that could potentially happen and build this foundation of predictive information. Apart from that, if we look at restoration practices which we find, mangrove restoration is a very common practice in Sri Lanka, rightly so, because there is such great potential for climate change to be for the impacts of climate change to be mitigated, making use of restoration activity. This sort of maps which we have generated and the sort of information generated from this INVEST model, it can allow us to identify which regions of the lagoon which we should reforest. If we were to extend this to other lagoons of the country, we can identify then which lagoons will be least affected by the rising sea levels, which will be most affected by rising sea levels, and then we can pick and choose and make sure that the reforestation practices which we carry out are those that will actually benefit us in the coming 100, 200, 300 years. Thinking long term in this manner, it's extremely important, and we have to look beyond this sort of myopic decisions which we have sort of been making up to now. With that in mind, I will conclude. These are just some special selected references, and thank you very much for your time and attention.
Thank you very much, Mr. Kotagama. A very interesting and very informative presentation. And that you have talked about the blue carbon, because it's a kind of thing that we have to pay attention nowadays. And we know that the carbon dioxide is going to get increased. So I think that the audience has many questions to ask you or clarification. So dear audience, that the forum is over to you for questioning and also for answering session. Please unmute and raise your questions. Any questions? If there are no questions, Mr. Kotagama, may I raise questions? May I forward the questions to you?
Yes, of course.
It's very interesting because that you have used a model, and may I know that in your model that whether you have found out the land loss due to the sea level as well as the mangrove cultivation. So both conditions have you considered in this model, or that separately you considered that?
No, so it would be one extent, but then we group the land area depending on the different types of land use land classes. So it's like whatever the area, and then within that area, what are the different types of land use classes which are going to get affected by inundation.
Yeah, because with the inundation, we are losing land, and same times you are talking about the carbon sequestration. And
Yes.
So have you considered that losing land, as well as are you suggesting that we have to grow more lands, more mangroves, except in those lands?
Something along those lines. So the thing about it is, like, there's no point really growing mangroves if that area is eventually going to be subjected to inundation. The main purpose of what we wanted to really try to understand was which areas of the which areas of the lagoon are being subjected to inundation. And if we were to look at mangrove reforestation or whatever as a purpose of mitigating climate change, sequestering carbon, then should we continue to just sporadically plant these plants wherever we wanted? But rather, I mean, when you have an understanding of which areas are being subjected to inundation, it's very easy for us to pick and select the areas which will give us the best results.
Yeah, however, that the inundation is getting slowly because it's 50 years,
100 years, and then
so therefore that yeah, we can use those lands, even those are going to inundate after 100 years, so the 150 years. So may I know that within 50 years, like 2000, let's say 2050, right? Okay, yeah, 2050.
Yeah.
So what's the situation? Have you considered it? Because in Sri Lanka nowadays we are considering that the zero carbon in 2050. So they are going to have a roadmap, and they do so many research. The country is going towards a zero carbon in 2050. So what kind of contribution you can have or you can give for such kind of a roadmap? So such kind of situation.
So the thing about two things, one being the thing about 2050 is that with the predictions that we have, it wasn't data that is available for us to look at because we don't have sea level rise data in the country within the Chilaw Lagoon or locally. So we had to rely on what IPCC had given on the global sense, and that was for 2100 and 2100 to 100 and 150. Given that sea level rise is a slow process anyway, so we have to be mindful of that as well. Then the second point I just wanted to say is just like, what about beyond 2050? Because it's sort of unrealistic to expect that we will achieve zero carbon by 2050. just given the circumstances and the way things are going. But it's also important to look beyond that. It's not that we can get to zero carbon by 2050 and then we can maintain it in that way. I mean, what we've been seeing so throughout is that with time, there has been more technology, more emissions. So if we were to actually achieve net zero by 2050, that would be exceptionally great. But then what about beyond that? And how do we maintain it in sort of like a sustainable manner?
Yes, thank you very much. And so my next question is actually, I'm interested of your research. That's what I'm asking this question also, that we know that there are many mangroves types or species. So have you, your group or your research groups have selected a particular mangrove to be grown in Sri Lanka, or the set of series or or number of mangroves to be selected to grow, to absorb more carbon? Atmospheric carbon?
Yeah. So we obviously weren't able to look at which species would be absorbing the most amount of carbon, just given the time frame and the resources that we have. But then again, with the replanting thing, it's not just about growing one type of mangrove, because inadvertently what you'd be doing is having a monoculture then. So the next one, the presentation after this 133, we looked at the diversity of mangrove communities and then correlated it to the biomass carbon. And then, I mean, should I go into that now, or do we want to wait for the next one?
Right. Okay. The next presentation is also from Mr. Kottagama. So may I introduce that presentation, also the presenters. Thank you very much, Mr. Kottagama, for your good answering for discussion session. And so let's move to the next presentation. That's the number ID 133. The research title is Floristic Diversity and Distribution of Biomass Carbon of Selected Areas in the Chilaw Lagoon, Sri Lanka. The authors are O.W. Kottagama, K.A.R.S. Pereira, D.D.G.L. Dahanayaka, and S. Somaratne. So the presenter is Mr. O.W. Kottagama. The forum is over to you.
Good morning, everybody. My name is Odato Kottagama. I will be presenting on the floristic diversity and distribution of biomass carbon in selected sites of the Chilaw Lagoon. So among all the different types of ecosystems which we find associated to coastal environments, one of the most distinctive ecosystems which tends to stand out is the mangrove forests. Mangroves are found distributed throughout the tropical regions of the world. We know that they're restricted predominantly through the coast to the coastal belts of both islands as well as continents. So Sri Lanka is no different to this, and being an island country, throughout its coastal belt, particularly concentrated in lagoons, we have abundant growth of mangroves. And mangroves fulfill vast amount of ecosystem services, from filtration to coastal protection to all these sorts of different community benefits. They are all provided by mangroves. But what we find is that not all the mangroves, not all the lagoons throughout the island are equally well studied. Particularly the Chilaw Lagoon, it's one of the lagoons which are the least studied, and the literature and studies which are present on the Chilaw Lagoon, they tend to show a large amount of gaps in the studies and the data which they have been producing. So this study in particular, we wanted to evaluate the diversity as well as distribution of biomass carbon within the mangrove forests of the Chilaw Lagoon. We expect that the sort of information which we can generate from this study has the potential to be applied to different sorts of conservation as well as climate change mitigation goals. So the objectives of this study, predominantly there are two objectives. One was to determine the species diversity in the Chilaw Lagoon and also try to compare it with other lagoons through the use of published information. And secondly, we wanted to determine the biomass carbon that is present among each of those species and understand how it is distributed among the different types of species. In line with those objectives, the methodology which we utilized in order to carry out this study is indicated here. It's very straightforward. So we selected and only did the study to a particular region of the Chilaw Lagoon. We were unable to extend the study to the entirety of the Chilaw Lagoon just because of the lack of resources and just under the conditions at which we were carrying this study out. So we focused on the mangrove islands and the mangroves on the upper portion of the Chilaw Lagoon. So once we had identified these regions, we established belt transects, trying to capture as much diversity as possible within these regions. So we established 4 belt transects, which we then divided into 10 by 10 meter plots, and we carried out surveys identifying different species of mangroves. Now for the purpose of comparative analysis, so to quantify the diversity, we calculated the Shannon-Miner diversity index. So that included 2 parts, obviously the species richness as well as the species evenness. That is given here through these equations. So taking the information which we had obtained from our surveys, we applied it to these equations and we determined both the species richness as well as the species evenness among mangrove forests. Next, in line with the second objective of the study, we wanted to identify the biomass carbon, and for that purpose we first had to determine the above ground and below ground biomass. To do so, we took measurements of each mangrove tree. So we calculated the diameter at breast height by measuring the circumference of stems. All stems which were greater than 5 centimeters were measured within the transects which we established previously. We converted that circumference into the diameter and then applied it to allometric equations, standardized allometric equations, and we calculated both above and below ground biomass. Thereafter, by multiplying that value by a factor of 0.56, we were able to calculate the total biomass carbon. And here we have the different types of allometric equations which we utilized. Where it was possible to apply equations for specific species, we applied them that way. And for all other species, we applied the common equation. So when we move on to the results, the table that is indicated here is a summarization of all the results which we have obtained with association to the different types of species encountered, the individuals we encountered, the number of those individuals, as well as the biomass or carbon contribution. So Bruguiera cylindrica was the most abundant species, where 78 individuals were identified, and Avicennia marina, only 30 individuals were identified. That's the second highest. Species such as Bruguiera sexangula and Psilocarpus granatum were very rare and were found, only 1 individual of each were found throughout the surveyed sites. Of particular note is when we take into consideration the contributions of biomass and carbon, Avicennia officinalis, even though we only identified 23 individuals, contributed to 29.21% of the biomass or carbon contribution. But when we look at Bruguiera cylindrica, of which 78 individuals were identified, only 33.5% of the biomass or carbon contribution is provided from Bruguiera cylindrica. So the reason for this, which we were able to identify, was that the individuals of Avicennia officinalis which we found were much larger in size. They were older plants, so obviously they would have a capacity to store more carbon in comparison to Bruguiera cylindrica. So when we look to the diversity indexes, both species richness and species evenness was calculated. In the Chilao Lagoon, the species richness was 1.09 and evenness was 0.67. This is considerably lower, except for in the case of Puttam Lagoon, where Nigamuni and Subasinghe indicate that the species richness is 0.76. However, in comparison to Nigambo, Manditivu and Arali lagoons, the species richness is much higher, which is considerably understandable given the extent of the Chilao Lagoon, the mangrove forest, which is one, extremely small, and secondly also the level of disturbance which we observe in the Chilao Lagoon. So here the pie chart that is shown depicts the percentage contribution of each species to the biomass or the carbon. This is the information which was initially indicated in the table. So for ease of understanding, it has been included in the form of a pie chart here. And as we mentioned earlier, Bruguiera cylindrica contributes the largest with 33.5%, whereas Avicennia officinalis contributes 29.21%. So species such as Psilocarpus granatum, Numenstera racemosa, they contribute the lowest because the abundance of those species was also low. But as we mentioned, Avicennia officinalis stands out because even though it contributes 29.21%, which is very much close to 33.5% that was provided by Bruguiera cylindrica, we only identified 23 individuals of Avicennia officinalis, whereas 78 individuals of Bruguiera cylindrica were identified. In closing, just some key factors to sort of summarize the finding of the study. So the key finding which we were able to identify was that the diversity of mangrove species in the Chilao Lagoon is considerably high. This is important, especially considering that the lagoon is a very small lagoon and it also is subjected to a large amount of disturbance. So the degree of disturbance which we observe in the Chilau Lagoon is very high and comes about from the close interaction between communities as well as the expanding impacts of prawn farming. So the Chilau Lagoon, we know over time, was a very popular site for prawn farming, but as a result of this, large extents of mangrove forest have been cut down. This is a direct threat on the diversity of the lagoon, of the mangrove species in the lagoon, especially when we find that many of the species which we encountered, not only were they abundant, but there were also species that show a high degree of threat. So when we take into consideration the threatened status of species such as Bruguiera cylindrica, which we've encountered 78 individuals, is an endangered species. Likewise, Psilocarpus granatum is also an endangered species. So the species that are found in the Chilau Lagoon, not only is the mangrove forest rich in species, but it also is subjected to a large amount of threat. The biomass carbon information which we find in relation to the diversity can help to really point the direction towards effective restoration and reforestation practices. We know that restoration and reforestation of mangroves is a very popular practice in Sri Lanka, not only for the conservation of mangroves, but also for the conservation of the coastline, to protect it against erosion, and also to support fisheries and local communities. But restoration practices must be pragmatic. We have to take into consideration all these different factors, and one of the important factors to consider is diversity. We want to have restoration which at the end of the day will somehow reflect the situation, the natural situation which is present on the ground. So if we were to just take one species and then grow it in the lagoon, invariably what we are doing is generating a monoculture. So baseline information and understanding of what the situation is on the ground at the present with association to the diversity of different mangrove species is very important, so that we can make decisions regarding how and what species to be included in restoration practices. You also, like, in the sense that we also want to avoid introducing species which may not be local species that are found in that lagoon. So in this sense, localized studies are very important, and they can help us to prioritize which species should be subjected to conservation, and even in restoration practices, which species should be included in restoration and reforestation. And thank you for your time and attention.
Thank you very much, Mr. Kotugama. I think that you have given the answer to this research for the questions I have raised in the previous session. However, that I'd like to know about I want to have few clarification. Before that, I'd like to hand over the session to the audience to raise questions if you have. Any questions from the audience? If there are no questions, since the research is so interesting, I'd like to know about a few more things. Would you please explain that the difference between these mangroves and for the carbon sequestration and also the terrestrial forest? That do you have any idea? Because we know that in Singarajo there are many terrestrial forests in the country, but we heard that the mangroves are really good for the carbon absorption. So would you please explain regarding that?
Yeah, so from what I understand, mangroves, the carbon that they store, it's not only in the plant, but it's also below ground. Because of the conditions, the water retention and the anaerobic conditions that are present, all the sort of litter and the wood which falls and gets accumulated in that area, it tends to remain there undecomposed for much longer periods of time than in rainforests. In rainforests, we know that nutrient cycling is so rapid. I mean, soil in rainforests tend to be depleted with nutrients because the absorption of nutrients is so quick, carbon included. So in the case of mangroves, I mean, why we find that there are such great ecosystems for carbon sequestration is primarily because of that reason, that you have such a large below ground carbon reserve, just because of the conditions that are prevalent. Yeah.
Okay, thank you. Any question from the audience once again? You have mentioned that, of course, that the combination of species of mangroves would be better than, because otherwise it will be the monoculture. Anyway, it's a monoculture if we are talking about the mangroves, but however, by combining many species of the same mangroves would be good for rather than having a one species. Like my question is now Trilindrica that you mentioned and also Officinalis. That two species, we have seen that Officinalis is given a much more attention and higher biomass than the Cylindrica. So can we have those two combinations together, or what kind of suggestion do you have? Because you have done research on many species in the same land, but you have selected. So would you please explain regarding that? Yes.
Sort of depends on what your conservation goal would also be. If it was primarily and only to mitigate carbon and capture as much carbon dioxide from the environment as possible, maybe based on what we have found, it could be those two species. But that again, I'm not committing to that as species that are best for carbon capturing, because we haven't done that as a study. This is just the situation that is present when we did the research. But I mean, now with climate change and all the environmental problems that we have, I think we're beyond looking at solutions that try to answer one thing only. It's really important to look at it in a multifaceted way, where you're looking at not only capturing carbon, but also preserving biodiversity, and you're ensuring that there's function of whatever the community requires from the lagoon. So I think it's quite difficult to answer that as just saying these two species only. You have to look at the broader picture. And I mean, generally we know that when you have more diversity, any ecosystem tends to be more resilient. So if we were to just, maybe if we wanted to look at only carbon capture, then just whatever species that we find may capture the most amount of carbon. But on a more sustainable scale, I feel like we want to try as much as possible to mimic what naturally exists. But that also will obviously possibly change, especially with like salinity and inundation and all these kinds of things that could potentially take place with climate change.
Thank you. There are many questions. Can I ask now? But whoever, that is the time also getting over. One question I'm interested on, that other than the species what you have studied or what you have collected or what you have seen in this selected study area, are there any species, endangered species or losing species in our country, in our coastal line?
There possibly might be. I think they put out a new red list in 2020. I think they evaluated all the different threatened species. So, I mean, we only found 8 mangroves in Chilau Lagoon. So
in Sri Lanka, in other lagoons in Sri Lanka, I'm telling you, okay, Trincomalee or wherever that, okay,
yeah, there must be.
Yeah.
I'd say
any
that
author can answer for this question. Yes. Any of your authors can answer my question?
Yes, actually, I'm Roshan here.
Yes.
This mangrove area is actually very, very important because that is located in intermediate zone. And this is run under low salinity. Most of the species are in low salinity. The other mangrove areas, most of them are in dry conditions and high salinity. This is very, very important area regarding the location. That's why we selected this area. Okay, thank you.
Okay, thank you, Dr. Roshan. And thank you, and all the best for the research group. And thank you very much. So let's move to the next presentation. That is ID number 137. The research title is efficacy of compost made of food waste with organic amendments for environment-friendly agriculture. The authors are W.A.S.R. Fernando and T.K. Veerasinghe. The presenter is W.A.S.R. Fernando. The forum is over to you.
Good afternoon, all of you. My research is efficacy of compost made of food waste with organic amendments for environment-friendly agriculture. I'm W.A.S.R. Fernando, and I did this research with the assistance of Professor T.K. Veerasinghe. Agriculture is the main occupation in Sri Lanka, and we have a little problem with agriculture. We have limited amount of lands, and we are using the same land again and again for agriculture, and the lands are being depleted with nutrients. But composting is a good answer for that, and we have found that there are so many benefits of compost. According to Dalzell et al., it increases agricultural output, it improves long-term structural stability, it improves moisture retention of the soil. And also, if we use compost made out of food waste, there are added benefits like it reduces food waste being disposed in landfill, and also it helps food industries to manage their wastes. But there are issues of compost too. According to Ayla et al., it has pathogens and it has a low nutrient status. There's a long duration of composting. There's a long mineralization duration and also odor production. The most recently found, according to the IPCC, the problem of compost is the greenhouse gas emissions. So the solution has also been given by Barthod et al., telling that the negative aspects of composting can be limited by adding organic, inorganic or biological substrate to the composting mixture. This has been researched by various researchers, but mainly for inorganic and biological substrates. For organic substrates, biochar and mature compost have been tried. So I wanted to try food waste as an organic amendment to see whether the greenhouse gas emissions and also the limitations of the compost can be reduced by using it. The significance of this research is to present a quality compost with an organic amendment for the sustainability of the agricultural system. And the aim was to develop a good quality compost by adding different organic amendments to food waste and to improve the sustainability of agricultural systems in an environmental friendly way while enhancing soil functions and plant growth and reducing emissions. The objectives were to report the quality of the compost made with different organic amendments based on physicochemical parameters as a potting mixture, to evaluate the performance of different compost types on growth parameters with and without amendments, to investigate the potential of using organic amendments in reducing greenhouse gas emissions from compost for an environmentally friendly agricultural system, and to find out the best type of co-compost as a sustainable product. Food waste was collected from home as banana peels, eggshells, onion peels, papaya peels separately, and they were co-composted into as 1:1 ratio, and the quality of the compost was measured using physicochemical parameters such as total nitrogen, total phosphorus, total potassium, organic carbon, organic matter, C/N ratio, pH, EZ, and moisture content. Then 6 seedlings of Capsicum annum each were planted in potting mixtures with a 1:1 ratio of co-compost, topsoil, and sand, with a positive control of food waste compost and a negative control with no compost. The performance of the 6 treatments were evaluated by measuring the plant height, number of leaves in the plant, number of buds, number of pods, and the average weight of pods. Also, the carbon dioxide emissions was in the 6 treatments were measured by absorbing it to calcium hydroxide and then doing a titration. We used Excel Microsoft Office 2013 to find out the descriptive statistics and for graphical data representations. And we used Minitab version 3.1 for statistical results and discussion. As you can see, the nitrogen, phosphorus, and potassium content is a little bit low for egg and onion amendments. But when I compare the NPK contents of banana peel, they seem to be at the approximate value which Reddy has given for NPK, that is 0.5%, 0.27%, and 0.81%. Even compost made with amendment of banana peel showed higher amounts for organic carbon and organic matter content. And also this can be explained by the higher carbon content in them according to Anhongwe, Unge, and Nyatke. And pH values of compost showed values to the neutrality, and all of the amendments showed values approximately from 6 to 8.5, which has been suggested by Hogg et al. And also compost with banana peel amendment showed a higher moisture content. Moisture contents are important in keeping microbial populations active because sufficient moisture is required for microbial nutrition and transport. Performance of compost in increasing plant height and number of leaves per plant. As you can see from the graphs, the purple color line is going much underneath than the others. The purple line is a T0 treatment where no addition of amendment was done. So these results are in agreement with Mercy et al., who stated that height of fenugreek plants was higher in fruit peel powder applied to soil than the control. But when we did the statistical analysis, the T2 treatment, that means the banana peel amendment, showed a significant growth rate compared with the treatment without compost. So why is that? That can be explained by the nitrogen and potassium content that were high in the banana peel amendment in the earlier slide. So nitrogen has been found to be an essential element for plant growth, and also potassium is also an essential nutrient that helps in biochemical and physiological processes influencing the plant growth and metabolism. Performance of compost in yielding flowers and pods. According to this table, you can see the T0 treatment has not given any yield of flowers and pods, but the others have done. Treatment T3 with the eggshell amendment showed the highest number of flowers per plant and also pods per plant, while the highest average weight of pods was shown by plants with the treatment T2, that is banana peel amendment. When we take the earlier table with the banana peel amendment, the compost had higher amounts of phosphorus, potassium, and organic matter content. Phosphorus and potassium content has been found to increase crop growth by Eidemir and Innes. So according to that, we can conclude that banana peel amendment has shown a higher yield because of its higher nutrient content. Carbon dioxide as a greenhouse gas emission. This graph shows the amount of carbon dioxide emission within the incubation period of 35 days. So all the emissions showed a milligram of 3.5 to 4 of carbon dioxide emission per day per 1 kilogram of soil after 2 weeks of incubation. This behavior is similar to the observation in Ray et al., where carbon dioxide emissions spiked following the organic amendment regardless of organic amendment types and the application rates. In the current study, the carbon dioxide emission was low compared to the research studies. The T0 had the least carbon dioxide emission, and that is mainly due to the absence of organic amendment to the soil. This is in correlation with Ray et al., where cumulative soil carbon dioxide emissions from all the amendment plots were higher than those from respective unamended plots. According to Searle et al., he has shown that the biochar made with banana peel seems to be more efficient reducer of greenhouse gases than the normal peel. Conclusion. Compost made with banana peel amendment was identified as a high-quality soil amendment which can be used as an organic fertilizer in agricultural systems. When considering the different compost types, plant treated with the compost made of banana peel amendment showed a significantly higher growth rate and yield. Thus, we can conclude that compost made with banana peels perform well compared to other amendments. All the amendment compost types had a higher emission of carbon dioxide than the soil without compost. Therefore, the compost made with banana peels can be recommended as an additive, soil conditioner and potting media for sustainable use, which could be used. Also, can be enhanced as biochar. Thank you very much.
Thank you very much, Ms. W.S.R. Fernando, for your very informative and nice presentation. And now the forum is open to the audience to ask questions. If you have any, if you want any clarifications, please raise your questions and you can join with the discussion. If there are no questions, Ms. Fernando, I'd like to ask a question from you that is there any reason to select these particular items like banana peel and eggshell and onion and papaya that when you are selecting your particular composting items?
That was because that was the most common stuff, and I needed to find a higher content which can be like had to make a lot of compost. So most common stuff that can be used, and according to literature, they have been used mostly. Those are the main food items that have been used in various literature also. So I decided to go for those stuff.
Okay. I'd like to know about when you are making your compost. We know that eggshells are kind of dry item and also onion leaves. Okay. Yes. I hope that you have collected only the onion, whatever the cover
peels.
or the
yes.
peels. And those are, we can consider as dry items. That means moisture content is really low. But when we talk about the banana peels as well as papaya with a high moisture content. So how do you make your compost based on this?
When I'm making the compost, I have to consider about the amount of water I'm adding. I have to, again and again, I'm making the compost, I have to add water to keep the water retention. Now, when I mixing the compost, during the composting procedure, when I'm mixing the soil, I had to add more water for egg and eggshells and onion peels, mainly eggshells, to keep the moisture content there. Yes.
Okay. How long that it took to make the compost?
About 46 days, but had to check the pH values until they became neutral. I had to keep them because I have to make sure that the composting is almost done, completed.
I have doubt with these eggshells because we know that it's deteriorating of eggshells are getting, I don't know whether it's take time.
Eggshells, it was actually grinded and was added man.
So right, okay.
Yes, as powder.
Right, okay. So with that, you have found out the banana peels are the best type item.
Yes.
Right. Okay. May I know that the contents you have added for the soil, the amount of the same amount by the biomass or like the volume?
Volume 1:1 ratio.
If seems that the eggshells volume is, but you have to collect the eggshells because others are with the moisture content when you consider about the volume, but in the eggshells and onion leaves or onion, whatever the cover or that the peels, that the volume
Volume, yes, what volume collection.
Volume collection and the density and all are going to get changed when it's going there. But I hope that you have, I have seen that you have considered the mass later on, that you considered per soil mass.
Yeah, amount
how much,
of emission, yes, was done.
emission.
Yes.
Right, okay. Yeah, thank you. Any other questions from the audience? Ms. Fernando, my last question is that how you are going to apply those things in the real world? Any idea that
now we know that,
and all?
yeah, this is the most crucial problem in Sri Lanka. No, we had this huge problem whether we are going to go for organic compost and not. Actually, I started this before that problem, actually, selected the topic. So actually, I think banana peels is good, but we have to again go for another research to check the amounts we have to include, the concentrations, the masses, amounts, and we can do a proper research about how much amount we need to put and how much yield then you will get, and then we can apply it to the agricultural system.
Okay, thank you very much, and also all the best for your future research.
Thank you.
Thank you much for all the authors for this ID 137. Thank you.
God bless.
Thank you. Let's move to the next session, next presentation. That's ID number 139. The title is Production and Investigation of Starch-Based Bioplastic Reinforced with Coconut Fiber Composites. The authors are K.N. Samaraheva, SNCM Dias, and J.I.K.G. Jailath. The presenter is K.N. Samaraheva. Forum is over to you. Thank you.
Good afternoon. My topic is Production and Investigation of Starch-Based Bioplastic Reinforced with Coconut Fiber Composites. Let's move to the introduction. Global per capita plastic consumption is 11 kilograms per year. Among developed nations, United States has per capita plastic consumption of 139 kilograms per year and Europe has 65 kilograms per year. Single-use plastic items released to the environment as waste without recycling cause unbearable waste load. For example, sachets, plastic bags, boxes, and cups. When comparing the usable and degradation lifetime, it is surprising because we use the content of the plastic item within very short period of time comparing to the degradation lifetime. Pollution, environmental pollution due to plastic waste and taking long time to degrade has become a global problem. So, numerous solutions have been proposed. One is to replace synthetic plastics from degradable bioplastics. So, this figure 1 shows the life cycle of some plastic items. Let's move to the significance of the study. Usually, we use a plastic straw within 1 or 2 minutes, but it takes 200 years to degrade. This seahorse usually lives 3 to 5 years. You can see the skeleton of this seahorse grows around this plastic straw. So this little creature has to live its entire life cycle with this plastic straw as its body part. And we use a plastic bag once, but it takes 1,000 years to degrade. Sealfish usually lives 25 to 30 years. So this plastic bag causes death to this creature. Let's move to the problem statement. Plastic became an essential commodity in day-to-day life. So, due to its durability, it leads to a waste problem after end of their useful lifetime. So, numerous solutions have been proposed. One is to replace synthetic plastics from bioplastics. Let's move to the aim of the study: to produce alternative biodegradable bioplastics to substitute petroleum-based synthetic plastics. And let's move to the objectives: to produce coir fiber reinforced plant-based bioplastics using sweet potato, and manioc, and to determine and compare the tensile properties of bioplastic coir fiber composites. Let's move to the literature review. These are some past related research studies related to the bioplastics. Let's move to the methodology. First one is extraction of starch from sweet potato, and manioc. And next is the determination of starch percentage of 3 grams. And next, production of bioplastic sheets with 3 fiber concentrations. And next, determination of cleaner production potential of 3 bioplastic types with 0.0 fiber concentrations, and determination of tensile strength of 3 fiber concentrations for 3 bioplastic types. These are the chemicals and apparatus, and data was analyzed by Minitab statistical software. This plate A shows the extraction procedure of sweet potato starch, and plate B shows wet and dried sweet potato starch, and plate C shows the apparatus and chemicals for the production process, and plate D shows gelatinized translucent gel of starch intermediate process during the production process. And this is the sample preparation for tensile strength experiment. 25 samples were tested. This is the equation for determination of starch percentage of sweet potato, and manioc. And for determination of tensile strength, Newton balance was used, and the weight needed to break the bioplastic sheet was measured, and 25 samples were tested. And figure 4 shows the process flow diagram of the production of bioplastic sheets according to the cleaner production strategies. Let's move to the results and discussion. This figure 5 shows the average percentage of starch for 300 grams of 3 samples of potato, sweet potato and manioc. We can see manioc contains highest starch percentage and potato contains lowest starch percentage. This plate 1 shows produce bioplastic sheets according to the cleaner production process. Manioc containing bioplastic sheets have the least transparency when comparing to the potato and sweet potato containing bioplastic sheets. This table 1 shows the quantified material balance and water balance for sweet potato based bioplastics, and table 2 shows the total cleaner production potential. And this table 3 shows the cleaner production potential for potato, sweet potato and manioc based bioplastic sheets. And we can see manioc is the most profitable when comparing to the potato and sweet potato based bioplastic production. This plate 2 shows the produced bioplastic sheets with and without fiber. We can see by fiber reinforcement the transparency of bioplastic sheets were reduced. Let's move to the tensile strength. Figure 6 shows the average tensile strength for 3 bioplastic types with 3 fiber concentrations. we can see manioc contains highest tensile strength for no fiber concentrations and fiber with 2.5 gram fiber concentrations and 5 gram fiber concentrations. And also we can see with fiber incorporation, tensile strength increase in 3 bioplastic sheets when comparing to the 0.0 fiber concentration fiber reinforcing. And let's move to the conclusions and recommendations. Manioc contains higher starch percentage and potato contains lower starch percentage. And manioc-based bioplastic is most profitable production compared to the sweet potato and potato-based bioplastic production. The transparency of the bioplastic sheets reduced with increased in higher fiber percentage. The tensile strength of 3 bioplastic types gradually increase with addition. Let's move to the recommendations. Edible packaging material can be produced for food items from starch-based bioplastics by using only edible ingredients. And also we can try bioplastic with waste food items. Waste food materials such as banana peels and other food peels, potato peels like that. And also production of bioplastic sheets according to the cleaner production strategies are more environmental friendly. Biofiber reinforcement can produce bioplastic with high strength to improve durability and quality. These are my references. And thank you very much, and I would like to give my special thanks to Madam Chamila Dias and Madam Kumudika Chailat for proper guidance, supervision and support during the study period. And also I would like to thank all my teachers. And as human beings, we should be responsible of other living beings on this earth, because we have only one planet and we should protect this earth for our future generation. And also we should use this planet in sustainable way, and we should not harm these small creatures by our new innovation and by our consumption. So as human beings, we should think twice about this matter. And thank you very much.
Thank you very much, Ms. K. Samarheva, for your very excellent presentation. And you have drawn our attention for a special thing, microplastics and the killing of many animals in the world. And the forum is over to the audience for question and answering session. If there are no questions from the audience, Ms. Samarheba, may I ask a question from you that I'd like to draw your attention to the durability of plastics, what you have prepared bioplastics. That have you done any research, or are you planning to have research on that side?
Yes, madam. Actually, I would like to try for bioplastics with waste material in future, without using edible materials, because edible materials can be used for such as sausage, vegetarian sausage casing like that. And so other than that, I would like to try for bioplastics with waste material.
Right. Thank you. My next question is, I want with clarification of your calculation, what you have done about the total cost, and it's around over 125, yes, 12 millions, and it's around total cost is 12 million, and also that the total waste profile is somewhat a higher number. I think it's 151 million, if I'm correct.
Madam,
So how did you do that calculation?
yes, madam, I was calculated considering during a day, 10 bioplastic sheets were producing, and for a month, 20 working days are considering, and from that, for 12 months, for 1 year, I was calculated that calculation, and the problem is solid residue from filtrate was very high. So because of that, the cost is high, because we are taking solid material can be used for other products if we are considering as a production process, because that solid material here, that's considered as a waste material, solid residue from filtrate. So that can be used for cereal products, biscuits, like that. That is why next time I would like to try for production of bioplastics from waste material. For only edible food materials, I prefer this producing bioplastic from edible ingredients.
If you are considering the waste materials, then you have to do the killing microorganisms and all, whatever the pathogens. However, it would be somewhat costly, or anyway that you can find out a better manner. Yes.
The cost is high because of solid residue from filtrate is high in the output side. That is why the cost is high. That is not a real waste, madam. We can use that waste to produce cereal products and other food materials. That is why the cost is high of production.
So out of these three items, like a potato and sweet potato and manioc, that you prefer to have potato or the manioc, because we know that manioc have some experience or some ability for like a carcinogenic. It's a opposite of carcinogenic material. So are you going to use manioc or other than the rest, like a potato or sweet potato?
When considering the cost, manioc is the best for the production process. I was not considered about the carcinogenic properties in this study.
Actually, it's not the carcinogenic, anti-carcinogenic properties of manioc, isn't it?
Yeah. Then I prefer manioc for the production process from this study.
Right. Any question from the audience? If there are no questions from the audience, that I'll wish my all the best for the research team, Samaraheva, Ms. Dr. SNCM Dias and Dr. J.I.K.G. Jaila, and wish you all the best. Thank you for your presentation. Thank you.
Thank you.
And with that, we are moving to the last item of our session, the environmental sciences, that the ID number 160. The title of the presentation is Suitability of Granular and Powdered Activated Carbon as an Absorbent for Desalination. The authors are D.S. Samaraheva and SNCM Dias. The forum is over to you, Ms. Samaraheva, the presenter of this presentation.
Good afternoon. Today I am going to present our research paper for the International Open University Research Symposium 2022. Our paper ID is 160. The topic of our research paper is Suitability of Granular and Powdered Activated Carbon as an Adsorbent for Desalination. These are the content of our presentation. Let's move on to the introduction part. The world we live in today is so commercialized that people simply forget about the natural resources and environment. Therefore, the world demand for drinking water has significantly increased since the world's population has tripled in the 20th century, which is expected to increase by another 40 to 50% by 2050. As an island surrounded by the Indian Ocean, it is a great opportunity to purify seawater in Sri Lanka. According to the Earth's water budget, oceanic water is the large sum of water source in the Earth. When considering the annual spatial distribution of rainfall patterns in globe and Sri Lanka, they are not homogeneous. When comparing the arid zones and semi-arid zones in Sri Lanka, such as Hambantota, some parts of Uva province, Northern province and North Central province, they are getting less than 120 millimeters of annual rainfall per year. They are annually facing the droughts. Therefore, as a developing country which is surrounded by sea, low-cost environmental feasible method is more suitable for desalination for the water scarcity in Sri Lanka. Therefore, drinking water scarcity in Sri Lanka. Therefore, this study is based to develop such method using adsorption technique and membrane filtration process. These are the objectives of the study. The main goal is to find a feasible solution to desalinate seawater to be used for drinking purpose as a solution of water scarcity problem in Sri Lanka. The specific objectives of the study were to find a low-cost solution in remove salinity of seawater by adsorption isotherm and to evaluate the salt removal efficiency of different activated carbon filter media. Let's move on to the methodology part. Figure 1 shows this sketch diagram of the procedure. According to that, the raw materials were used for this study was coconut shell, bamboo, watermelon peel and sawdust. First, preparation of granular and powdered activated carbon. All the raw materials were dried under the sunshine and separately heated these samples under 600 degrees, 700 degrees and 800 degrees in an oven, and then washed from hydrochloric acid in order to get out from the impurities. Then again dried in an oven for 60 degrees for 3 hours and then sieved using 2 mm sieve in order to get the granular activated carbon and 0.2 mm sieve in order to get the powdered activated carbon. Next, preparation of sodium chloride aqueous solution in related to the same concentration of the natural seawater sample sodium chloride concentration. Batch experiments were done varying the parameter of adsorbent weight for the granular activated carbon samples and powdered activated carbon samples separately. Column experiments were first done to the granular activated carbon samples separately using the granular activated carbon layer sandwiched in between 2 sand layers. Then done to the same procedure for the powdered activated carbon samples. Again done the column experiment using granular activated carbon layers sandwiched in between 2 hydrilla layers, and then again 3 layers were sandwiched in between the 2 sand layers and separately done to the granular activated carbon and again done to the powdered activated carbon. Next, adsorption studies were done using the synthetically prepared sodium chloride aqueous solution in order to prove the Freundlich isotherm. Eigenvalue calculated in order to prove the absorption sites are favorable. Finally, data was statistically analyzed using Minitab software. The data statistically analyzed using pooled t-test and paired t-test. Let's move on to the most important part of this study, that was results and discussion. According to the table 1, basic characteristics of different activated carbon was shown there. According to this, proximate analysis fixed carbon and elemental analysis carbon and the ash analysis silicon oxide samples, we can clearly see that fixed carbon elemental carbon and silicon dioxide is relatively very vigorously high in the sawdust when comparing to the other 3 activated carbon sources. Therefore, sawdust has the highest favorable absorption characteristics of carbon analysis were done using the Fourier transformation infrared spectrophotometer. Next, move on to the table 2 that shows the schematic images of different activated carbon from scanning electron microscope. According to that, sawdust has many small pore spaces with large surface area. Therefore, absorption capacity when comparing to other 3 carbon sources very high in the sawdust because here are large pore spaces but surface area is too small. Here very limited pore spaces and here very large pore spaces with very limited surface area. Let's move on to the iodine value. According to the calculation of iodine value using the sodium thiosulfate titration method, iodine value was very high in the sawdust comparatively to the other activated carbon sources used in this study. Therefore, absorption capacity of the salt ion is very high in the sawdust. Next, move on to the proven of Freundlich isotherm data. According to the Freundlich isotherm proven data, Freundlich isotherm was proven according to the Freundlich isotherm constant was very high in the when we compared to the other 3 sources of activated carbon, that was very high in the sawdust because this is very highly proven the y equals mx plus c graph here. Therefore, the equilibrium concentration of the equilibrium constant and the Freundlich isotherm constant is very high in the sawdust. Therefore, sawdust has the consecutive layer model and the favorable sorption sites. According to the batch experiments were separately done to the different carbon sources of granular and powdered activated carbon, the adsorbent weight was very high in the sawdust. When comparing granular activated carbon, it is very high, and powdered activated carbon, it is somewhat low. Therefore, the conclusion is granular activated carbon sawdust has the highest adsorbent weight or the salt removal capacity. Let's move on to the column experiments. As I mentioned in the methodology part, after statistically analyze the column results using paired and pooled t-tests, results can be summarized that salt removal efficiency was relatively high in sawdust and the salt removal efficiency was relatively high in granular activated carbon sawdust. The capacity can be increased using hydrilla verticillata because hydrilla is very highly adsorbed the salt ions. Therefore, the efficiency is nearly 83%. Let's move on to the conclusion part. Desalination processes effectively with the granular activated carbon. Due to the availability of most pore spaces, sawdust activated carbon adsorption capacity is vigorously high when compared the reverse osmosis technique, which was the highest efficiency desalination technique in the world, adsorption isotherm technique and membrane filtration process, which were used in this study has shown a considerable low cost and high efficiency in desalination process. The adsorption data followed by the Freundlich isotherm confirming the consecutive layer model to unlimited sorption sites. This was useful to the physical sorption of the seawater because seawater is a combination of salts. Iodine value is the indication of capacity to adsorb the salts. In this study, activated carbon of sawdust have the most favorable and highest iodine value. Therefore, it also proves that sawdust is remarkably absorbent. Column study saw membrane filtration process. The ratio of amounts 20 gram used was was in this study sand to hydrilla to granular activated carbon of sawdust to hydrilla to sand equals 1 to 1 to 1 to 1 ratio. This is the highest desalination effect ratio. Efficiency was 83%. Adsorption technique of desalination shows strong positive linear relationship. When using granular activated carbon of sawdust, the removing capacity of salt was 73%. Water samples were analyzed through atomic absorption spectrophotometer after the treatment of granular activated carbon of sawdust have the significance and considerable decrease in chemical parameters and cations and anion concentrations. Therefore, granular activated carbon of sawdust have a significant level of salt removing capacity in relation to this study. Thus, this study shows adsorption technique and membrane filtration method using granular activated carbon of sawdust was highly effective, environmental feasible way to desalinate the seawater. Let's move on to the recommendations. Increasing the amount of granular activated carbon in the column will increase the efficiency in desalination process. Increase the diameter of the column will increase the rate of desalination. Thus, this water purification or desalination process can be simply identified as the clean production technology with the concepts of industrial ecology, industrial symbiosis, greening the supply chain. This can be developed up to a filter using a motor inside the column. Rotating filter will increase the adsorption to the membrane. Therefore, it will be low-cost, high-efficient, environmental feasible method to desalination. References of my study. I would like to give my special thanks to my co-author, Madam Chamila Dias, and I would appreciate her guidance given to me. Finally, I would fondly appreciate the opportunity given to me in this International Research Symposium, and I would like to thank all the organizing committee of this International Research Symposium. I want to mind blow you through this quote: "The cure for anything is salt water, sweet tears of the sea," by Isaac Dynson, because salt water is the very important water source in the world for humans and animals for this water scarcity problem. Thank you and have a nice day.
Thank you very much, Ms. Samaraheva, for your nice presentation. Now the forum is over to audience for question and answering session. Any question from the audience? If there are no questions from the audience, I have a clarification to get from Ms. Samaraheva. In your presentation, you mentioned that you have put in your items like your raw materials to the oven to get the GAC, granular activated carbon, and also that activated carbon. So 700, have you used these 700 degrees in an oven? Or you
in a first I have separately used the clean, and then I used the oven to dry it.
Is it the oven or the muffle furnace that where you have used? What's the instrument you have used for this item? This heat 700 or 700?
Furnace I have used, but 60 degrees I have used an oven.
600, you mentioned 600 or 60 degrees?
Finally 60 degrees. Before that I have used 600 in a brick kiln. In
what?
Earlier,
brick kiln. Yeah. So have you know about the temperature about 600? Are you sure that the temperature was that amount?
It is very highly amounted. I have used a high amount, and I used separately to measure that using a temperature up to that level, but exactly related to that level. Approximately, I used that level.
Okay, my next question is about your isotherms. So
Yeah.
you said that friendly isotherm is fit for your results, but I have seen in your presentation from your graph that if you have used a different isotherm, that would be better because especially the sodas will be given the better fit because the items are like whatever the data is here and there, and so there are many isotherms that you can select, and if you have tried those things, would you may get a better results. What is your idea?
Langmuir isotherm I didn't get because of the consecutive layer model. The sorption, it is a combination of salt seawater. Therefore, sorption is not favorable for Langmuir isotherm because it has a monolayer model, but here not the monolayer model. That is why I have used the friendly isotherm model.
That is not fitting very well, no? So I think it's better if you carry out more research in this area, you better to find out that the best option or best fitting model for your research.
Okay.
And okay, you mentioned about that the practical application of this one. So we know about the sodas anyway, that we heard that the powdered activated carbon are the best one. Let's say from the coconut shell is the best activated carbon, as it gives that the hard coconut husk. So that's what we experience. So, but here you are mentioning that the sodas is much better because of the pores. My suggestion and my recommendation is that if you are going for that, better to find out your the best fitting isotherms and try to find out the mechanisms what's happening there, that whether by using that the best fitting that you can understand having the physiochemical or whatever the absorption over there, and by doing that you can find out the best solution for using your method. Right.
Thank you, madam.
Yes.
Thank you.
There's another question I have regarding this about your application, that how you are going to apply this one in future. Because we know hydrilla is somewhat very rare things, and it's kind of a plant in the sea. So why you have used hydrilla? Because what's the condition you have made this hydrilla into your research?
Yeah, they are also absorbing the hydrilla, absorbing the salt ions, and they are decaying, not decay, they are absorbing and decaying that plant. Therefore, I have used that hydrilla as a layer in that filter. It also give the more efficient to the filter rate. That is filter. That is why I have used the hydrilla as a layer in the column.
I hope you have not done without hydrilla in your column, don't
no,
you?
I have done without hydrilla also. When I used the hydrilla, there's a slight color change occurred with using the hydrilla. Apart from that, it is high efficient. Without hydrilla, it has given 73%. With hydrilla, it has given 83% of removal capacity.
Slight change. Okay.
Yeah.
My last question, that if you are using this one, have you considered about the grams or whatever the per gram that the removal percentage or the efficiency of removal of salt?
Yes, madam. First, I have done the batch experiments varying the adsorbent weight. There I used 1, 2, 3, 4, 5 grams of the adsorbent, and I kept in the rotating shaker. And then secondly, I used changing the volume of the seawater sample. Then I have done the research varying the parameter of kept time of the rotating shaker. Lastly, I have done the kept time of the rotating shaker. Varying those parameters, I have done the batch experiments. In this paper, I only focused about the adsorbent way.
My question is that how many grams do you have to use for removal of salt from 1 liter of water?
1 liter of water, nearly 20 grams, 20 grams nearly we want to use 20 grams of granular activated carbon in the filter medium.
That means after 1 liter goes, that you have to replace your
20 grams
media once again.
Yeah.
So then it's good that you have done that part, and please do the calculations to see that how applicable it is. Because if 20 grams fulfilled by the 1 liter, then you have to, because for drinking or whatever the purposes, we want to have many liters. So how many grams you have to use per day, so that you have to calculate. But it's, however, that is a very good research, and you have tried your best. And see my recommendations about to find out the best isotherm, to find out that the best to understand the adsorption mechanism. And however, I wish you all the best and for your research group. Thank you, Ms. Samraheva.
Thank you, madam.
Right, with that one, we are going to conclude our session of environmental science. And thank you for all participants as well as the audience, and all the presenters who has done well, and congratulate for their research. And so with that, with all the congratulations to all authors and the presenters, and wishing a very good evening for all the audience, we conclude our session on environmental science in IHours 2022 International Open University Research Sessions. Thank you.